and Chemistry Kids. So I think I've got some kind of arrangement working where it actually works for me. So we're looking at proteins. So proteins are in most of our body structures. We're going to kind of look at them as food, but we have to think of food as something we need for our bodies. Otherwise it's just a thing that we eat to keep energy going and to build things and all that. It's a point. So we need it for hormones, it's enzymes, so it makes things happen. I'm trying to remember my example. It's completely escaped my head, but there was one thing where if we didn't have enzymes, it would take like 1,000 years for it to happen in our body, and it happens really super quick. It makes antibodies so we can fight viruses. It makes structures like muscles and skin and hair and muscles and skin and hair and things. You're kind of not going to be expected to know what proteins make what. They're going to say the protein blah blah blah, the protein sequence of this. No, what would they say in the question? They would give you the protein and why it's used, but they might then ask you to talk about something in terms of the bonding maybe. But really this is just a kind of an intro. So they're all going to have hydrogen, nitrogen, oxygen and carbon. Some have phosphorus and sulfur, which makes them have slightly different properties. The plants make their own proteins. We have to get it from food, so we either eat the plants or we eat something that has eaten the plant, or we eat something that's eaten something that's eaten the plant. So that's where we're getting our energy from. So this might be a bit, will this be year 10 biology? I think we did this anyway. So proteins are made up of amino acids. So we get our amino acids. We've got about 200 that we know of, but there is 20 that are in the human body. So those are the ones we're kind of concerned with. The rest we're going to be concerned with if we're looking at other things, but these are humans, so we're kind of going to. So we would know of them, we see them in the book and in the data book, named secondary or alpha amino acids. This is because we have an amino group and a carboxyl group bonded to a secondary carbon. So the second carbon. What does this mean? Well, it means we have a general structure that looks like this. So we've got an amino group, we've got an R group, which is going to change depending on our amino acid, and we've got a carboxyl group. And we can see that, so we can see that in our data book. Like this. So I got this from that Jamie Kennedy guy. I'll put the link in the lesson. So this is really useful for when you come into the exam and you go, what is all this? I can't remember. So that first, not reading time, after that reading time, going through this and just labelling. And once you've done it, maybe practiced a few times at home. He's got the whole thing labelled. I have not reinvented the wheel. I've used exactly the same colours he has. The only thing I did was add this bit at the top. But what's really good is they've given you these groups. So your amino group and your carboxyl group are here and here. So they're not changing in their structure. So you really don't have to, don't have to think, but you don't have to think about what that's. Then we've got our properties up here. So that's non-polar. This one's polar, non-basic. This one's acidic. This has got our sulfur, so cysteine is an interesting one because it makes these little bridges. And I just went through and did that, and it took me 2 minutes, and I had a copy up on my screen. You won't have a copy on your screen in the exam, but it will be really useful for you to do this. Like practice a few times, and then when you come into the exam, either you'll have to do it or you won't have to do it because you've got it so kind of memorized that you won't have to do this annotation. But by having this annotation in your head, it will simplify it, and it means when you are jumping between questions, your brain can kind of go back to your kind of summary. This is almost like a summary of the information. So that's our data. Oh, I don't need to get rid of it because I'm going to use it. Right. So our amino acids, they can be essential or non-essential. So essential, and this has completely escaped my head, and it shouldn't, because essential is the ones, the essential are the ones that we need to eat. So we can't make those ones, so we need to eat them. I think leucine is one. Why can't I remember what this is? Leucine is one. 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 lysine, valine. Yeah, so those are ones that we need to eat. So they might be added to things. Those protein shakes that people drink will have lots of these ones. These non-essentials we can just get from, we can make. So because we can't store these, we have to keep taking them. These ones, we can make them from other things. So we can break things down, build them up. There is a list in the JAG Plus of what they are. I've not seen any questions when they've asked you to identify, which one is an essential, which one is a non-essential. But you will have to say what maybe the difference is. So one thing about this is you might also get asked about, well, what this food is compared to this food. If we've got some essential food, amino acids in there compared to our non-essential, it's going to be a better food source. So things like lentils and nuts and certain seeds have these essential amino acids in, so they make them like a better choice if you're not eating meat and things, and better for the environment and all those kind of things around that. So I have got these amino acids, but obviously it's not just some glutamic acid wandering around my body doing the job. So I'm going to make strings. It's probably the best way to start thinking about them. Strings of these amino acids building together to make first dipeptides and then polypeptides. We've got 4 levels of structure that you need to know about. It's a weird I think this is the thing they're going to get rid of, because I don't quite understand the level they want you to know. I've looked at some examples, so I'm going to go in and give you all of the information I can. We'll try some questions and you'll figure out what that level looks like, because I think there's lots of tricksy things in here that I think are beyond the scope of the study design, that are going to trip you up if you start going down that rabbit hole, because this is a lot of this structural stuff for proteins is a uni thing. So and it's a tricky uni thing because it's hugely interesting. Right, so okay, so we've got dipeptides and polypeptides, and they both form the same way by a condensation reaction. Right. So we go from 1 amino acid to get 2 amino acids, and this is going to depend completely on the amino acids you have, so which 2 or more you have, and the alignment. And then we've got polypeptides, which is going to be many, normally about 50 and more. And then that's also going to depend on your amino acids you're given and the alignment of them. And when you do this type of question, you have to show that it's a long, ongoing chain. So you have to remember, I read that more for myself than for you, to show the bonds at the end. So we'll start with our dipeptides. So we have 2 of our, we'll start with the generic amino acids. I've put my pencil over there. Right, so we have our generic amino acid. And you will see this R and H kind of either way. I don't know, does it make any difference? But it's probably better to think about it at the top, because that's where they've put this R group in the data book. Oh, I didn't give myself enough room. Give yourself enough room. Write it properly. Right, so we've got those 2, and they're coming together in a condensation reaction. So they've got a condensation reaction, and what we're getting out is water. So we've got this group here. Oh, I'm not drawing it very well. Right, let me draw it easier for you to understand, rather than squashing it in there like a crazy person. Right, so So we've got our two generics here, just some, and it's going to be the same for whatever. So this is kind of a good way to remember it in your head. So we've got condensation, so we have water involved. So this water here is being taken out. So this water here. So what we end up with is some water from there. And then we have this kind of being joined together. So then we have a should be better. Try and make better lines to show your understanding than me. Do as I say, not as I do. And then we have this section in the middle here. Which is the peptide link. Tink link. So this bit here is our peptide link. Let me do that one again because I don't like it. So whatever we have, we're going to have this peptide link. I've seen a few questions where they've said they've given you like this or even longer. They've given you like 3, and you have to identify the peptide links, because that's not something you can get just by looking at the data book. They might give you 3. Oh, where's my question gone? Oh, it's gone off the board. Right, give me a sec. I was going to do a question from the exam. Let me disappear. Right, so the question was from 2013. It was saying spiderwebs are very strong and elastic. Spiderweb silk is a protein that consists mainly of glycine and alanine residues. Assume these amino acids residues alternate in a spider web. Draw a section of the spider web protein that contains 3 amino acid residues. So it's got to have 3 glycines and alanines, and we're going to assume that they are alternating. So my 2013 spider question. So we must have or gly. Right, so it's going to be either of these 2 options because you're showing that alternating, and you're going to draw the section of the spider web that contains at least 3 amino acid residues. So I probably need another page because my drawing is too big. So I need, let's see, gly. So don't have to remember. So we've got alanine here. Oh, let me move this up. Here. So GN CH. I should draw that out properly. I'm going to bond them in a bit. And then we've got glycine. Oh, they've given you nice small ones to do at least. And then we've got another these NH. Right, this is a step you're not going to need to do because you're going to just skip to the next step, but you might need to do this roughly to get your head around it. It depends on how you are comfortable with this. So we've got these 3. I've sketched them out, so now I've got my I would make sure you're always using pencil for this, because we will stuff up. So we're getting rid of this one and one of these hydrogens here. Would have helped if I'd have drawn it out. And we're doing the same here. So we've got a and then we've got NH, E, H, H, and H down there, CH. Oh, I should put that down there. Keep it consistent. CH3. Right, so that's that question. So I've got my 3 here that I didn't really need to draw, but it was quite helpful in thinking about how these were bonding. And then I've got my 3 peptides, my 3 amino acids bonded together. The name of the bond between each amino acid is the next question. So this is my bit here, my bit here, and these are called peptide links. What type of polymerization reaction occurs in the formation of spider web links? Well, we said before it was a condensation reaction. Okay, then we've got a question you're going to need to do next, and then next. So that's an exam question. That's 1, 2, 3, 4 marks there. So you got 2 for this. I'd probably get it there. That's a bit dodgy. I'll get 1 for saying it was that, and then 1 for that. So 4 marks, pretty easy. Even with this bit, it actually didn't take me very long, and these 2 were quicker than these 2 marks were quicker than that, making that 2 marks. So remember, it's a marathon with 20 bits, so there are bits that are going to be quicker when we do our working out of the total time. Okay. So now I'm going to make another video where we look at our primary, secondary, tertiary and quaternary, I can't say that word, structures of proteins.